In this video, we will talk about the neural crest cells. Do you know what are neural crest cells? As we have discussed during neurulation, neural crest cells appear in the crests of the waves of the ectoderm that are moving towards each other. In next step, the neural tube has formed, and neural crest cells move away. Now, let's have a short overview of role of neural crest cells. Neural crest cells become parts of a wide range of organs and structures, and differentiate to form a variety of different cell types. For example, they will form much of the peripheral nervous system, skeletal parts of the face, and pigment cells in the skin called as melanocytes. Migration of these cells is well organized and an important part of the normal development of much of the embryo. Now, let's discuss the migration of neural crest cells, where time period is from day 22. If we focus at this point, this is neural tube. This is mesonephrinic duct. In male embryos, the mesonephric ducts give rise to the ductus deferens, epididymis, and seminal vesicles. The ductus deferens connects the testes, where sperm is produced, to the urethra, allowing the transportation of sperm during ejaculation. The epididymis provides a site for sperm maturation and storage, while the seminal vesicles produce seminal fluid, contributing to the composition of semen. In contrast, in female embryos, the mesonephric ducts undergo degeneration and do not contribute significantly to the development of the reproductive system. Instead, female embryos develop Mullerian ducts, which give rise to structures such as the fallopian tubes, uterus, and upper part of the vagina. This is mesonephros. The mesonephros, also known as the intermediate kidney, is an intermediate stage of the kidney development that occurs during embryonic development. It is the second of three sets of kidneys that form sequentially in vertebrate embryos. And this is gut. Above gut is the dorsal aorta. Now, let's discuss the two groups of trunk neural crest cells. Migrate in different directions. The migration of neural crest cells begins in the cranial end of the embryo, shortly before the neuropores of the neural tube close. Although they soon become interspersed amongst the cells of the embryo that they are moving through, they can be tracked in the lab with cell labeling techniques. Now, two groups of trunk neural crest cells migrate in different directions are: one group of neural crest cells migrate dorsolaterally around towards the midline ectoderm, and other group of neural crest cells migrate ventrally around the neural tube and notochord. Now, let's discuss the destinations of neural crest cells. As we have discussed, development of the central nervous system. From neurulation, the central nervous system continues to develop as the cranial end of the neural tube dilates and folds to form spaces that will become the brain, the remainder of the neural tube. Caudal to the first four somite will become the spinal cord. Neural crest cells have different destination that will be discussed now. Now, let's discuss the destinations of neural crest cells. Neural crest cells will differentiate into melanocyte. Do you know what are melanocytes? Well, melanocytes are cells of neural crest origin in the human epidermis. Melanocytes are well known for their role in skin pigmentation, and their ability to produce and distribute melanin has been studied extensively. Parafollicular cells. Do you know what are parafollicular cells? Well, parafollicular cells, also known as C cells, these are a special subset of cells within the thyroid gland that secrete calcitonin. It is a hormone necessary for the regulation of calcium. Schwann cells. Do you know what are Schwann cells? Well, Schwann cells are a type of glial cells of the peripheral nervous system that help form the myelin sheath around the nerve fibers. Myelin sheath of the neuron. A Schwann cell envelopes and rotates around the axon forming myelin sheath. Now axon is myelinated. Chromaffin cell. Do you know what are chromaffin cells? Chromaffin cells are neuroendocrine cells found mostly in the medulla of the adrenal glands in mammals. Make epinephrine and norepinephrine. Conotruncal septum. Do you know what is conotruncal septum? Conotruncal septum is the septum that divides the conus cordis into the outflow tracts as well as the truncus arteriosus. Connective tissue, bones of face and skull. Ganglia. Do you know what is ganglia? A ganglion is a collection of neuronal bodies found in the voluntary and autonomic branches of the peripheral nervous system. Ganglia can be thought of as synaptic relay stations between neurons. The information enters the ganglia, excites the neuron in the ganglia and then exits. Odontoblasts. In vertebrates, an odontoblast is a cell of neural crest origin that is part of the outer surface of the dental pulp, and whose biological function is dentinogenesis, which is the formation of dentin, the substance beneath the tooth enamel on the crown. Adrenal medulla. Pia mater. Pia mater is the inner layer, closest to your brain tissue. And arachnoid. Arachnoid is the middle layer. Now, let me ask a question first. Then you can easily memorize the destinations of neural crust cells, or structures derived from neural crust cells. All of the following are derived from neural crest cells, except adrenal medulla, arachnoid, dura mater, or melanocyte. I will answer this question later. Remember that MPSC is a type of exam, so mnemonic to memorize. The destinations of neural crust cells, or structures derived from neural crust cells, is MPSC, goal, M4. Melanocytes, P4. Parafollicular cell, S4. Schwann cells, C4. Chromaffin cells. Connective tissue, bones of face and skull. And conotruncal septum, in heart. G4. Ganglia, O4. Odontoblasts, A4. Adrenal medulla, L4. Leptomeninges, which include pia mater and arachnoid. So, the question was, all of the following are derived from neural crest cells, except adrenal medulla, arachnoid, dura mater, or melanocyte. Now, do you know the answer? Yes, the answer is dura mater. So, this was all. We're excited to dive deeper into the fascinating topics of the brain and spinal cord. As we explored in our previous videos, the development of the central nervous system in an embryo is a complex and intricate process. One of the most crucial aspects of the CNS is the formation and maturation of the brain and spinal cord. In our upcoming video, we will delve into the intricacies of these remarkable structures, their development, and their vital roles in the functioning of the human body.
Ever felt overwhelmed by the traditional way of learning medicine? Enter the future of education with digital learning. Whether you're refining your knowledge of biochemistry or immersing in cell biology, it's all there. Understanding complex medical subjects has never been easier or more efficient with engaging, interactive content. Welcome to the new age of medical education. Learn anywhere, anytime, at your pace. Powering your medical journey. sqadia.com